Okay, so let's do lecture 12 of Molbio, and I'm going to specifically be talking about RFLP, or restriction fragment length polymorphism. One thing you really need to know is that we use this in missense mutations. Yes, and so the whole idea of RFLP is that you have your sample, you isolate the DNA, you do PCR, and then you incubate the PCR fragments with a restriction enzyme. Now, what would be the point in putting it with a restriction enzyme? And this restriction enzyme will cut the DNA fragments, blah, 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 blah. So basically, you can either cut with RFLP, you can either cut wild type, or you can cut allelic variant of alleles. So for the protocol that we did, you have this case. So for the analysis of this allelic variant, RFLP method is applied. PCR product is 475 base pairs long, and here they have just pointed out what the primer is, and etc. Now they have also told us that the wild type in this allelic has a G, which is this one, and allelic variant means that it has been exchanged to an A. So this G would be an A. So yes. Now when we use RFLP, we use an enzyme to cut the DNA in the place where we think there might be an allelic variant, or we could cut a wild type. But let's just follow this protocol and try to answer the questions. Find which one of from given restriction endonucleases will be able to cut the PCR product. Okay, so here we have CC and GG, GG and CC, GATC, CATG. Okay, so which one of these will be able to cut the PCR product? And what we can know is that what we can know from the exercise is that this is an allelic variant. So here they have given us the wild type. They've given us the wild type, but they have told us that they have applied a restriction enzyme to the allelic variant. This means that the restriction enzyme will go around and look for this sequence, not for a G, but for an A. Let me just write that. So a restriction enzyme, its job is to just go around and look for these exact sequences. And when it finds these exact sequences in the DNA, then it will come in and then it will cut. So as we can tell here, they have put a restriction enzyme that wants to specifically cut an allelic variant. Now, if we look at these three examples that they've given us, CC and GG. Okay, if you put this restriction enzyme, it will go around and look for CC and GG in the DNA. Now, there is no such sequence here, therefore it would cut nothing. If we look at GATC, you can see that this restriction enzyme, its job is to go around and look for this sequence, and when it finds the sequence, it will bind to the sequence and then it will cut. Now, here the wild type has GGTC, okay, but they have told us that they have put it they want to cut an allelic variant. So therefore, this as restriction enzyme would find the GATC and then it would cut. So therefore, this one would be correct because we have this sequence. If the G is exchanged to an A, which means allelic variant, then it will come and it will bind. And CATG is just wrong. There is no sequence that is CATG here. So this would do nothing. So this is correct. Okay. So now that we have applied our restriction enzyme to this DNA sequence, what will happen is that it will cut the DNA into two different lengths. So that would mean if we have 475 base pairs, after putting restriction enzyme to the DNA will be cut. And when it's cut, then that will lead to us having 2 fragments of different lengths. Okay, so this means in an electrophoresis picture, and let's not think so much about this one. So this will be the RFLP electrophoresis. Okay, so this means let's look at patient number 1. We said that a healthy person, a person with a wild-type DNA, will have 475 base pairs. That's a healthy one, okay? Now, if we look at patient 1, we can say that he has approximately 475 base pairs. Nothing has been cut. So therefore, we can say that he is healthy or just wild type. His genome would be wild type. Okay, but we look at patient number 2. We can see that he has a fragment that is 475 base pairs, and then he has 2 fragments here that are of different length. What does this tell us? This tells us that he has one wild type allele and one allelic. Oh shit, I can't write. One allelic variant allele. So what has happened here is that for patient 2, we took his DNA, we did PCR, and then to all of these fragments that have been produced after PCR, we put a restriction enzyme that had been going around the DNA and then looking for this allelic variant, and when it found the allelic variant, it cut the allelic variant. And this means that it found it on one allele but not the other. So this is why we have 475 here, but we have different lengths of base pairs or fragment. And you can see here 253 and 222. In the case of allelic variant, restriction site is formed, obtaining 253 and 200 long fragments, blah 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 blah. So basically, if the restriction enzyme has found an allelic variant, it will bind and then it will cut, and then you will obtain these 2 guys. And this is what has happened here. So therefore, patient number 2 will have wild type but also allelic variant. So in one allele and in the other allele, it will have an allelic variant. Now, if we look at patient number 3, we can see that he has no 475 base pair allele, which means that on patient 3, he will have allelic variant on both alleles, because the restriction enzyme has found allelic variant on both alleles, and therefore it has cut both alleles, and therefore you only have this. And on patient number 4 and 5, you can see that both are wild type, because the restriction endonuclease has found nothing to cut. So that would be that. Now, I have told you in the beginning that you can use RFLP to either cut a wild type sequence or you can cut a allelic variant. Now we have talked, so if we want to know if somebody has allelic variant, we know that the sequence, let's say, is ACTG. Let's say that is the healthy. So a healthy person would have ACTG sequence, but a non-healthy person, or non-healthy but like they have an allelic variant, let's say for example they would have ACGG. Okay, so the allelic and this would be wild type. Yeah, so you would have ACGG. What you would do then is that you use a restriction enzyme, restriction enzyme, and this restriction enzyme would have the sequence ACGG, right? And this ACGG, if it finds the allelic variant, it will come and it will bind to it. This is how you use RFLP for allelic variants, which is what I just talked about here. But you can also use another restriction enzyme that finds a CTG. And so you find this restriction enzyme and you put it together with the PCR fragments and whatnot, and then it will find the ACTG and then it will cut it. So it will be exactly like this. So the result will also be an electrophoresis, but if there are fragments that are cut, this wouldn't mean that the patient has an allelic variant, but this means that the patient has a wild type variant. So it goes both ways, both wild type and allelic variants. Yes, and so this is the kind of exercise that we got on the protocol. There's another exercise on the self-assessment questions from lecture 12. So, results of RFLP analysis for a rare autosomal recessive allelic variant are shown in the gel electrophoresis picture. The restriction enzyme chosen for this test cuts variant allele, while the wild type allele is left intact by the enzyme. List the patients that blah 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 are heterozygotes. Okay, I don't know what just happened. Anyway, patients that are heterozygote, which would mean that they are either wild type, and they have allelic variant. So this means this one, and this one, and this one. Yep. So all of them would be wild type and allelic variant because we have one fragment that has not been cut, and then we have one allele that has not been cut, and then one allele that has been cut, and the same thing goes for all of them. And basically what has happened is that for patient 2, you put his DNA in a tube, you put a restriction enzyme. This restriction enzyme found an allele because it's looking for an allele, it's not looking for wild type. It looks for an allele, it found the allele, and then it cut the allele. And this happened for patient 2, 4, 6, 8, 10, and 11. Now, our homozygotes for allelic variant. So that would mean that this person has only allelic variant, which would be 7, 9. And you can tell that because they don't have any allele here. Would be affected by the recessive disease. That would also be 7 and 8, or 7 and 9, because, let's see here, it's an autosomal recessive allelic variant. So that would mean if it's autosomal and it's recessive, then that means that it does not have any wild type. And if it doesn't have any wild type, then it will definitely have the disease. And then has a wild type genome, and that would be number 1, number 3, 5, I think. Yeah, number 5. Yeah. So they will have a wild type because there has been no cuts and the patient is healthy. Yeah, so this is RFLP. If you have any questions, just email me. Thank you.